Hello everyone, my name is Egwom Onyinyechi from Bayero University, Kano in Nigeria. I'm here to make a presentation of our research titled an SVM-based breast cancer prediction model using linear discriminant analysis for feature extraction. Our research presentation is going to go through this following format. We have the introduction, the methodology used, the results, the conclusions, and the recommendation. Data repository containing medical record for patients on diseases such as cancers are gotten through experiments, observations, and prescriptions. Cancer as one of these diseases can actually be caused, or we said is actually an outgrowth or an abnormal growth of the cells in the body. It can be breast cancer, blood cancer, cervical cancer, and it is mostly caused by environmental factors, which are mostly result from stress and change in weather. And if not treated and detected on time, it causes early death, infertility, and hormonal problems. It is mostly common in low-income countries such as Africa and sub-Saharan countries in Africa. Now, breast cancer is one of the common type of cancer diseases. It mostly happens when there is a change in the breast tissue, which divides uncontrollable and results to lump. It ranks as one of the highest or major cancer in women, with a fewer cases in men. The World Health Organization in 2019 estimated that there are about 268,600 new cases of cancer, and with about 41,700 deaths. Now, a good number of researchers have worked to determine how to predict if a breast cancer cell is benign or malignant. A benign cell is said to be a non-cancerous growth, and while a malignant cell is said to be a cancerous growth. How do they try to dictate this breast cancer growth? Because early detection can actually lead to high survival rates. Now, the primary concern of breast cancer prediction is to correctly identify that a breast cancer cell is either too benign or malignant. A good number of researchers have reported the use of machine learning for breast cancer prediction. Machine learning, which is a branch of artificial intelligence, learns to predict new concepts from input data which are gotten from past experience. The learning process in machine learning can be unsupervised. Under the supervised learning, we have the classification learning, which ranks among the most common problem solver for supervised learning, since all data are being labeled. Feature extraction is a technique also used in breast cancer prediction. It is used at the pre-processing stage. It's a technique that reduces the dimensionality of the data by compressing it. It aims to reduce the number of features in a data set, create a new feature from the existing data set, and the new feature is a combination of. Previous studies have shown that using some machine learning techniques actually have lead to problems, such as the issue of using a single machine learning algorithm, model inability to choose a proper feature extraction technique, which can effectively reduce the dimensionality for better prediction. A feature extraction techniques being unable to handle large data, which could lead to overfitting and the model's instability to work through. This research aims to develop a model for breast cancer prediction using linear discriminant analysis, feature extraction technique, and support vector machine algorithm. The aim of our research work is to use a linear discriminant analysis feature extraction technique on the existing data sets for dimensionality reduction, design a model for breast cancer prediction that uses SVM classifier on the reduced data set, use the experimental data set to evaluate the performance of the proposed. The execution of our research, or the model to be used for our research, is going to follow through the following process. We have the pre-processing of data, where our data is going to be gotten from the University of California Irvine Machine Learning Repository for Breast Cancer, which has an instance of 699 patient record with 458 instances as benign and 241 instances as malignant. We have 16 missing values which will be computed with the media of each class, Then the linear discriminant analysis as a feature extraction technique is going to be applied on the benign tumor and the malignant tumor. Then we are going to have a reduced data set. The data set is now going to be splitted into the training and testing data, which a tenfold cross validation is going to be applied on the tests, on the training and testing data. Then the support vector machine as a classifier is going to be used on the tenfold validation data sets. Then the confusion matrix is going to be used to evaluate the performance of our data. The data to be used for this research has two classes, which is the malignant and the benign class, and 9 integral values. These integral values have an ID ranging from 1 to 10. These sample attributes that are going to be used are the clump thickness, the uniformity of cell size, the uniformity of cell shape, the marginal adhesion, single epithelial cell size, the bare nuclein, bland chromatin, the normal nuclei, the mitosis, and the class, which has 2 as a value for benign and 4 for malignant. Linear discriminant analysis is mostly used at the pre-processing step for machine learning application. It projects a feature space of n-dimension data into a smaller subspace known as K, and K, which is the new matrix, is lesser than the previous data size, that means -1 lesser, but it maintains the class discriminant, which actually still has a distinguished feature for the malignant and the benign class, but it tends to maximize the mean of each class and minimize the spreading within the class maximum. The support vector machine is a linear model for classifier. Its function is to select sample from all class known as the support vector and separate the class by generating a linear function that divides them using this support vector. Its objective is to find a hyperplane in an n-dimensional space in which the margin is maximized. The classifier model is going to be evaluated using the confusion matrix, which actually shows the accuracy of our model. We have the predicted class and the actual class, which has the true positive, the false negative, the false positive, and the true negative. Our model is going to be evaluated based on its accuracy, sensitivity, and specificity. Using the confusion matrix for the classification accuracy for our proposed model based on the tenfold cross-validation, the first experiment was carried out using the support vector machine without any feature extraction technique. The result shows an accuracy of 0.96, a sensitivity of 0.97, and a specificity of 0.98. By the feature extraction techniques, we have an increased accuracy of 98%, a sensitivity of 97%, and a specificity of 97%, which shows an increment of 0.02 from the previous result gotten without applying feature extraction technique. Now, when comparing this result to other research work that have been carried out using other feature extraction model, we came to realize that there was a significant increment in the accuracy compared to the work of Bizeng et al. That made use of K-means and SVM, which had an accuracy of 97.38%. Then the work of K. Sivakami had an accuracy of 92%, and Rajiv Bharat had 91.2%. In conclusion, in this our research, we got to know that using the linear discriminant analysis and support vector machine for breast cancer prediction model has shown a significant improvement in terms of accuracy, sensitivity, and as well as specificity when compared to the other approaches. For feature recommendation, we actually ask that other feature extraction technique can be used to further improve the accuracy of our model, and how good is this model going to perform when tested on other breast cancer data sets. Thank you for listening. At this, I've come to the end of my presentation.